my name is Dr. Chloe Puyu, and today we are diving into the world of droplet digital PCR, or ddPCR. This video is going to go step by step all the way from calculating reagent concentrations through to interpreting results from our real lab data live. So why ddPCR? ddPCR is extremely sensitive. So ddPCR partitions your samples into tens of thousands of oil droplets. Each droplet acts like a teeny tiny test tube where an independent PCR reaction occurs. After amplification, these droplets are scanned one by one for fluorescence, telling us which ones are positive and which ones are negative. Using this data, we generate a Poisson distribution through which we can then back calculate the actual number of DNA copies in our sample. So that means that ddPCR doesn't rely on standard curves. It gives you an absolute count of DNA. So this precision, this quantification makes it super applicable for cases like measuring viral loads, quantifying rare mutations, or tracking microbial communities and environmental samples. Here's a roadmap to this tutorial. Number one, we're going to introduce one of our research projects. It's kind of a case study to anchor ddPCR and how we go about things and why we've chosen this method. Two, we are going to calculate how much of each reagent to add, and really starting at square one of like what and how much, and how do you dilute from stocks, for example. Then we'll go to assembling all of those components together, generating droplets. Then fourth, we'll move on to thermocycling. And finally, we will read the plate and look at our real ddPCR data in real time and do some interpretation with that. So whether you're brand new to ddPCR, just brushing up on your workflow, or just curious about how it works, I hope this video helps demystify the process. With that, let's get started.
Today we are going to be using ddPCR technology to quantify infection intensity in wild zooplankton toads. We have the zooplankton hosts, specifically cyclopoid copepods, that ingest the parasite. The frequency of this occurrence is very rare within natural landscapes. So what we need is a super fine, super sensitive technology to be able to detect presence of parasites within the hosts. So just to give you guys a bit of a background of where these samples come from, we can think about these lakes where we actually got these samples. So imagine there's a bunch of zooplankton that occur in the water column. We're out here throwing toes, collecting animals. Those animals get concentrated into a sample. Within this sample, you have algae, you have zooplankton of varying genera, cyclopoids, calanoids, daphnia, so both target host and non-host species. From there, we're going to be extracting DNA of the entire sample. We've designed two sets of primers and probes in order to detect both the host and the parasite in this ddPCR framework. So specifically, we have a primer probe set for cyclopoid copepods, and then we have a primer probe set for the parasite Schistocephalus solidus, which is a helminth tapeworm. Come along!
Today's question is, what goes into a ddPCR reaction? By a 2x master mix from Bio-Rad, and that contains the buffer, the polymerase, and the DMPPs that will produce the product. We also add primers. These are the same primers as you might use in regular PCR or qPCR. We do take into account the temperature, the melting temperature of the primers, because we want the probe primer to have a higher melting temperature than the amplification primers. So the probe primer is a fluorescently labeled primer that will bind to the middle of your PCR product. And then you have water to make up the difference of volume, and then your template DNA. Okay, so we are going to calculate what goes into our ddPCR reaction. The problem we have today is that we have a rather complicated reaction because we have two different conditions. We have a singleplex reaction and a multiplex reaction, so we need to take that into account. The singleplex reaction uses just a single probe. We're making a single application product and then looking for a single product with a single probe. In this case, it will be the Schistocephalus solidus, and we're using higher template concentration for that so that we have a better chance of finding very low amounts of the template DNA in our sample. And whereas the multiplex product, we'll be looking at the copepod DNA as well as the schistosomidas. And because these samples are basically pure copepod DNA, we will, if we don't dilute it a lot, we'll get too much amplification product and we won't be able to determine how much is there because every single droplet in the reaction would be constant. We'll have 36 Singleplex and 36 Multiplex reactions. The total reaction volume that we load into the ddPCR machine to make the droplets is 20 microliters. But because we want to have 20 microliters for sure, we will actually make a 22 microliter solution. So if we have 22 microliters of each reaction times 38 reactions will be our total reaction volume.
So does that include the master mix, the water? Like what does that, how does that relate?
Everything. We have 836 microliters, that's our final reaction volume. Because our master mix is 2x, that means that we have 418 microliters of our 2x master mix.
Did you do that in your head?
I did.
Nice.
We have 836 microliters, 900 nanomoles of our primers.
I hate this part.
Figure out how many microliters, we'll call that X, of a 100 millimole solution.
So how much do we have to dilute the stock solution to get it to our working?
Right. How much of our stock solution do we put into this 836 microliter total reaction volume? We have a factor of 10 to the third, 52 microliters of our amplification factors. So each reaction then will have 418 microliters of the master mix, 7.52 microliters of 7.52 microliters of primer 2, and then now we want to calculate how much of our probe primer is. So
So are you telling me we have forward and reverse primers, but we only have one probe?
in our Singleplex reaction, we only have a single probe of 8.36 microliters probe. For this experiment, 8.8 microliters. So in order to have 8.8 microliters of template in a 22 microliter reaction, that means that we want 13.2 microliters of our reaction mix plus 8.8 microliters gives us our 22. So we have 13.2 microliters of this stuff, and we have 38 of them. 501.6. So that is our number that we want as a final, so that when we aliquot 38 of these, then we 13.2 microliters in each tip. It's 60.2 microliters of water. Okay, so now let us calculate our reaction mix for having the multiplex reaction. So we need 7.52 microliters of our second primer 2 and our second primer 1.
So can all of these different primers and different probes exist within the same reaction?
They not only can, but they must. that is the beautiful process of ddPCR. Water.
And then you would add the same amount of template DNA to every single reaction, right?
That is correct. So I'm getting ready to set up our singleplex and multiplex reaction. So I've got two tubes, one labeled S in single, one labeled M and multi. I do have the water thawed. I have our master mix, and I have the various application types. Right?
Where's the DNA?
The DNA is over there.
That's right.
Away from this workspace. So
yes.
We have no chance of contaminating that into our master mixes, because that would be bad.
What about negative controls? What would I put in there if I'm not putting DNA?
8.8 microliters of the same water that we use in our master mix.
Are you guys going to use a positive control?
Yes, we are. We have a sample that is 100 copepods infected, each with one Schistocephalus worm, and we have DNA from that extraction that we will use as our positive control for both copepods and Schistocephalus.
Sounds like a cult about to kill us. This is like
Okay, so we are here with our PCR reactions set up, and we are going to start to make droplets. So this is the droplet generator. This is the droplet generating oil. This is a boat to put the oil in, and then we have a holder with a cassette cartridge clipped together, and then 70 microliters of oil will go in the lower chamber. Our 20 microliters of our PCR reactions will go in the center chambers, and then when it comes out of the machine, this upper row will have the droplets, with each droplet containing a PCR reaction.
So in every single well that you load, about how many PCR reactions are you expecting?
We typically get between 15 and 20,000.
So in one well, it's 20,000 PCR reactions.
That's correct.
Wow.
Okay, so now the droplets have been generated. We remove the gasket from the top of the cartridge. The upper row is cloudy compared to the other rows of the cartridge, and the cloudiness, those are the droplets. And so now we take everything that's in that upper chamber, which is about 40 microliters, and we transfer that to a PCR. So when you pull the liquid out of this chamber, all the droplets, in order to not destroy the droplets, you tilt the pipette tips at a slight angle, about 30 degrees or so, and then you lift up off of the bottom just a little bit so you're not pinching the droplets at the bottom of the well. You can see the droplets are at the top of the liquid mass, and then you eject it into the... Okay, so the samples are loaded into the PCR plate, and they're in the block for the heat sealer.
Alright, so now you have your sealed plate with your samples in it. The droplets are made, they're loaded, and that means it's time for PCR.
PCR, so we put the PCR plate in the thermocycler. You close the lid. You tighten the nuts. Okay, and bon voyage.
Bon voyage!
Now we're going to load our PCR plate into the reader. So you take the holder, pop off the lid, place the plate in the holder, red stripe up, press the button on top, the machine opens up, this slips in, make sure that it seats in the bottom, and press the button again.
Things that you should be mindful of when you're setting up the experiment that we'll touch on here. One is that we are measuring the absolute value. Two is that you are using that super mix, again, with no DUTPs. Very important. Also to remember, we have multiplexed reactions, so our first target is FAM. Yes, so that's our channel 1. We're going to put that as unknown. And channel 2, we're going to put on as hex.
Wow.
We'll be able to read the plate in real time and see the results in real time. You don't have to stick around to see a whole plate be run, but you could if you were very excited to see what happens. So this is an example of a reading from one of our results. So here the green channel is going to be the copepods, and the blue channel is going to be the schistocephalus. So you can see here there's proper separation between positives and negatives, which is what you're looking for. And you can see how many droplets there are. So there was about over 15,000 droplets that got processed for this one sample. We've just completed the entire run. It was better than we thought it was going to be.
Yeah.
That's a win. Let's go.
And then I know I did it a couple of times. But then eventually I figured out that, hey, yeah, I hope you captured that.
What?